Chapter 6, Cellular Respiration. Why cellular respiration matters. You can survive for weeks without eating and days without drinking, but you can only live for minutes without breathing. Why? Because every cell in your body relies on the energy created by using oxygen to break down glucose during the process of cellular respiration. You have something in common with this sports car here on the slide. You both require an air intake system to burn fuel efficiently. The metabolic process that produces acid in your muscles after a hard workout is similar to the process that produces soy sauce. The cells of your brain burn through a quarter pound of glucose each day. Biology and society getting the most out of your muscles. For athletes, their aerobic capacity is the limiting factor in their performance. So what is aerobic capacity? This is the maximum rate at which oxygen is provided or oxygen can be taken in and used by your muscles. Therefore, the heaviest exercise that your body can maintain aerobically. Endurance athletes must carefully monitor their efforts so that they maintain an aerobic pace over the long term. If you work harder than your aerobic capacity, then the demand for oxygen in your muscles will exceed the supply of oxygen provided by the respiratory system. In that case, the body goes into an emergency mode and switches from aerobic metabolism into anaerobic metabolism. Anaerobic means in the absence of oxygen, while aerobic means in the presence of oxygen. So this anaerobic cellular respiration, it breaks down glucose, which is the macromolecule or the fuel, very inefficiently, and consequently, lactic acid is produced as a byproduct. Sun is the ultimate source of energy for all life on Earth, and this energy is captured by the plants, the green plants, using the process of photosynthesis, and they convert the solar energy into chemical energy of sugars and other organic molecules. And then when animals feed on these plants, this is how energy is moved from one level to the next level. There are two types of organisms in this world, producers and consumers. Producers are known as autotrophs. They make organic molecules from inorganic nutrients by the process of photosynthesis. While consumers are known as heterotrophs, and they must consume the organic material and obtain energy by cellular respiration. And they obtain this organic material from the producers. Energy flow and chemical cycling in ecosystems between photosynthesis and cellular respiration. The molecular outputs of cellular respiration, that is carbon dioxide and water, CO2 and H2O, are the molecular inputs of photosynthesis and vice versa. While these chemicals cycle through an ecosystem, energy flows through entering as sunlight and exiting as heat. If we look at the process of photosynthesis, the raw materials required are carbon dioxide gas and water. Carbon dioxide is taken in by plants from the air via tiny pores, while water is absorbed from the soil by the plant's roots. Then once all the raw materials are collected, inside the leaf, the organelle called chloroplasts, they use light energy to convert these into organic molecules such as glucose, and a byproduct, oxygen, is released and goes back into air. On the other hand, cellular respiration in consumers will use the oxygen gas from the atmosphere, and it will be used to convert the energy to break down the bonds of sugars which are eaten by the consumers. It could be the molecules formed by the plants. And then the consumers, they produce a different source of energy called ATP, which stands for adenosine triphosphate. And this production of ATP during cellular respiration occurs in the mitochondria. And the waste products of cellular respiration are carbon dioxide and water, which are required by the producers or the plants for photosynthesis. Cellular respiration can be defined as the process by which Chemical energy is harvested from organic fuel molecules and then converted into adenosine triphosphate or ATP. This is usually an aerobic process. It requires oxygen and carbon dioxide is produced as a waste product. This is a multi-step chemical process and it involves enzymes to catalyze the chemical reactions at every step. What is the relationship between breathing and cellular respiration? Well, we breathe in oxygen from the atmosphere through our nose. It goes into the respiratory system. It reaches our lungs. Then from our lungs, the main transport agency in the human body, which is blood, it takes up oxygen, and then oxygen is transported to different parts of the body because each and every cell, every tissue in your body needs oxygen, and then that oxygen is given out from the blood, let's say to the muscle tissues, and then muscles use that oxygen. It goes into the mitochondria and it will be used to convert glucose, okay, that was obtained from the digestive system, and it's used to make ATP that will be required by the body. Carbon dioxide and water will be produced as waste products, and carbon dioxide is again taken up by blood and it will be taken to the lungs, and then we breathe out carbon dioxide. The overall equation of cellular respiration simplifies a great many chemical steps into one formula. You see here C6H12O6 is a glucose molecule. This combines with 6 molecules of oxygen, and this results in the formation of 6 molecules of carbon dioxide and 6 molecules of water. And this gives us approximately 32 molecules of ATP. There are 3 main stages of aerobic cellular respiration. The first step is glycolysis. The second one is citric acid cycle. And the third one is electron transport. Please keep in mind that each of these stages requires multiple chemical reactions. You can follow the flow of molecules through the process of cellular respiration in the diagram. The first stage of respiration, glycolysis, occurs in the cytoplasm of the cell, and the next 2 stages, the citric acid cycle and the electron transport chain, they occur inside the mitochondria of the cell. Now, if you notice that the bulk of ATP molecules are actually produced during the last stage, about 28 ATP molecules will be produced here, while the first 2 stages produce 2 ATP molecules each. During glycolysis, 1 molecule of glucose would be split into 2 molecules of pyruvic acid, and this step occurs in the cytoplasm. In the next stage, which is called the citric acid cycle or the Krebs cycle, here the glucose is completely broken down all the way up to carbon dioxide, and then the CO2 would be released as a waste product. The enzymes for the Krebs cycle are present in the mitochondrial matrix or the fluid which is present inside the mitochondria. Glycolysis and citric acid cycle generate ATPs directly and indirectly. There are very small amount of ATPs generated directly, 2 from glycolysis and 2 from citric acid cycle, while the amount of ATPs generated indirectly is way more because there are reactions that transfer electrons from the fuel molecules to a new molecule called NAD+, which is nicotinamide adenine dinucleotide. Okay? And then this electron transfer converts this NAD+ or NAD positive into NADH, which serves as a shuttle that can carry high energy electrons from one area of the cell to another. The third step of cellular respiration is electron transport. The enzymes required for this step are present in the inner membrane of the mitochondria. So the electrons that were captured from food by the shuttle in NADH, they are stripped of their energy a little bit at a time, and this keeps moving down in a step-like fashion, and finally these are combined with oxygen to form water. So electron transport from NADH to oxygen releases the energy that your cells will use to make ATP.
As this mountain biker heads up the trail, the breakfast he ate this morning is being burned to power his bike ride. His breathing rate increases as his leg muscles demand more oxygen to burn more fuel. Let's zoom down to where this fuel is burned, our cells. Here, the blood vessel on the left delivers fuel and oxygen to a single muscle cell. In cellular respiration, energy and fuel is converted to ATP, shown here as starbursts. Most ATP is made in the cell's mitochondria. ATP powers the work of the cell, such as contraction. Let's take a closer look at how ATP is produced from a molecule of glucose, our fuel. Only the carbon skeleton is shown to keep things simple. The first step is called glycolysis, and it takes place outside the mitochondria. To begin the process, some energy has to be invested. Next, the molecule is split in half. Now, the molecule NAD+, an electron carrier, picks up electrons and hydrogen atoms from the carbon molecule, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to reactions in the mitochondria. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced outside the mitochondrion. However, glycolysis has produced pyruvic acid, which still has a lot of energy available. Let's follow this pyruvic acid molecule into a mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, 1 carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that serves as the starting point for the citric acid cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. The carbon dioxide that you exhale comes from the reactions of cellular respiration. 2 ATPs are produced by the citric acid cycle for each molecule of glucose. At this point, only a small number of ATPs have been produced. However, more energy is available in the electrons that are being transported by electron carriers. While the citric acid cycle starts another round, let's follow an electron carrier to the next step in the process. Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondrion. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. The oxygen you breathe pulls electrons from the transport chain, and water is formed as a byproduct. The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondrion, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you've just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.
Let's get into a little bit more details about the different stages. Stage 1 is glycolysis, and glycolysis starts with the glucose molecule. The glucose molecule chemically is made up of 6 carbon atoms. So there are 6 carbon, it's a 6 carbon molecule, and during glycolysis it gets split into 2 pyruvic acid molecules, and each pyruvic acid molecule has 3 carbon atoms. And this initial split requires an energy input. Okay? 2 ATP molecules will be used for every 1 molecule of glucose. But at the end, glycolysis would produce a total of 4 ATP molecules. So the net gain would be 4 minus 2 equals 2 ATP molecules. Now the pyruvic acid that is formed, this is a 3 carbon molecules, they would donate high energy electrons to NAD plus and would form the shuttle NADH. Glycolysis is shown here in this figure. Before pyruvic acid can enter the citric acid cycle, and it has to be converted into a usable form. So the usable form is formed by the loss of a single carbon from each pyruvic acid molecule. So that single carbon goes out and forms carbon dioxide. Now the molecule that remains has only 2 carbon atoms and it's called an acetic acid. The acetic acid will get attached to a coenzyme called coenzyme A and it will form acetyl coenzyme A. Electrons would be stripped from these molecules and would be transferred to NAD+, and this would form more NADH. This figure shows how pyruvic acid is converted to acetyl coenzyme A, which can enter the citric acid cycle. The citric acid cycle completes the process of extracting energy from the sugar. It dismantles the 2 carbon acetic acid molecules all the way up to carbon dioxide. Carbon dioxide is a waste product that exits. So acetic acid, which is 2 carbon, joins another acceptor molecule that is 4 carbon, and the product is a 6 carbon molecule called citric acid, and the cycle is named after citric acid. For every 1 molecule of acetic acid that enters the cycle as fuel, 2 molecules of carbon dioxide would exit as waste product. This figure gives you the input and the output of citric acid cycle. Citric acid cycle keeps harvesting energy from the fuel to produce ATP directly and indirectly. In the indirect form, the cycle would capture more energy in the form of electron carriers such as NADH and FADH2. All the carbon atoms that enter the cycle exit out as carbon dioxide exhaust, and this 4 carbon acceptor molecule will be recycled. Stage 3, electron transport chain, which is a series of electron carrier molecules. At this stage, electrons that are obtained from glucose and other food molecules, they slowly fall and they lose energy at each step. In this way, respiration unlocks chemical energy in small steps, bit by bit, that cells can put to productive use. So electrons will be transferred from glucose in food molecules to NAD, and this would convert NAD to NADH. The NADH releases 2 electrons that enter the electron transport chain, and with each exchange, the electron gives up a bit of energy. This hard hat analogy shows that splitting the bonds of a molecule of glucose produces the energy that boosts an electron from a low energy state to a high energy state. So the big picture of all this transfer of electrons is a downward trip or slipping of electrons from glucose to NADH to electron transport chain and finally to oxygen. What is the role of oxygen in harvesting food energy? In cellular respiration, electrons fall in small steps from food to oxygen, producing water. NADH transfers electrons from food to an electron transport chain. The attraction of oxygen to electrons pulls the electrons down the chain. The molecules of the electron transport chain, they operate in the inner membranes of mitochondria, which is highly folded to increase the surface area. And because of the increased surface area, thousands of copies of electron transport chain can exist here. So the energy stored by the electron transport, a good analogy is something like the water behind a dam. The inner mitochondrial membrane not only contain the electron transport chains, but they also contain turbine-like miniature machines called ATP synthase, which are made of proteins. And this ATP synthase is used to generate ATP from NADH and FADH2. How does electron transport drive ATP synthase machines? Each electron transport chain acts as a chemical pump that uses the energy released by the fall of electrons to move hydrogen ions, or H positive, across the inner mitochondrial membrane. So if we look at 1 and 2, NADH and FADH2 transfer electrons to an electron transport chain. Number 3, the electron transport chain uses this energy supply to pump hydrogen ions across the inner mitochondrial membrane. Number 4, oxygen pulls electrons down the transport chain. 5th, the hydrogen ions concentrated on one side of the membrane rushes back downhill through an ADP synthase, and this action spins a component of the ATP synthase just as water turns the turbines in a dam. The rotation activates parts of the synthase molecule that attach phosphate groups to ADP molecules and generate ATP. The poison cyanide produces its deadly effect by binding to one of the protein complexes in the electron transport chain, marked with a skull and crossbones symbol in the figure. When bound there, cyanide blocks the passage of electrons to oxygen. This blockage is like clogging the outflow channel of a dam. Consequently, no hydrogen ion gradient is generated and no ATP is made. So cells stop working and the organism dies. Cellular respiration can generate a maximum of 32 molecules of ATP per molecule of glucose. But remember that the actual number may vary by a few depending on the organism and the molecules involved. Glycolysis and the citric acid cycle each contribute to ATP by directly making it. The rest of the ATP molecules are produced by the ATP synthase, powered by the fall of electrons from food to oxygen. The electrons are carried from the organic fuel to electron transport chains by NADH and FADH2. Each electron pair dropped from a transport chain from NADH or FADH2 can power the synthesis of a few ATP. Although we have concentrated on glucose as the fuel that is broken down during cellular respiration, remember that respiration is like a very versatile metabolic furnace that can burn other kinds of food molecules as well. So monomers from carbohydrates such as polysaccharides and sugars, fats and proteins can all serve as fuel for cellular respiration, and there is interplay between these pathways. This figure illustrates some metabolic routes for the use of carbohydrates, fats, and proteins as fuel for cellular respiration. Taking together, all of these food molecules make up your calorie-burning metabolism. There is another way in which food energy can be harvested, that is known as anaerobic respiration or fermentation. Some of our cells can work for short periods of time without oxygen, and this type of respiration is anaerobic respiration, which relies on glycolysis. So fermentation relies on glycolysis, which was the first stage of cellular respiration as we learned. And glycolysis does not require oxygen, but produces 2 ATP molecules for 1 molecule of glucose that is broken down to pyruvic acid. The main difference between aerobic and anaerobic respiration is the presence or absence of oxygen, but this gives rise to another problem. To harvest food energy during glycolysis, NAD must be present to receive electrons, and in aerobic conditions, NAD can be regenerated, but recycling of NAD cannot occur in anaerobic conditions because there is no oxygen. So NADH disposes of electrons by adding them to the pyruvic acid molecules which were formed by glycolysis, and this in turn produces a waste product called lactic acid. This figure shows fermentation resulting in the formation of lactic acid as a waste product.
All cells are able to synthesize ATP via the process of glycolysis. In many cells, if oxygen is not present, pyruvate is metabolized in a process called fermentation. By oxidizing the NADH produced in glycolysis, fermentation regenerates NAD+, which can take part in glycolysis once again to produce more ATP. The net energy gain in fermentation is 2 ATP molecules per molecule of glucose. Fermentation complements glycolysis and makes it possible for ATP to be continually produced in the absence of oxygen. There are 2 types of fermentation. Alcohol fermentation, which occurs in yeast, results in the production of ethanol and carbon dioxide. Lactic acid fermentation, which occurs in muscle, results in the production of lactate.
What causes muscle to burn after hard exercise? Fermentation in human muscle cells. When muscle cells consume ATP faster than oxygen can be supplied for cellular respiration, the conditions become anaerobic and muscle cells will begin to regenerate ATP by fermentation. The waste product under these anaerobic conditions is lactic acid. The ATP yield per glucose is much lower during fermentation, 2 ATPs, than during cellular respiration, about 32 ATPs. This question of muscle burn was investigated by a Nobel Prize winning British biologist named Dr. Hill. He developed a technique for electrically stimulating muscles dissected from frog and kept them in a laboratory solution. This is Hill's 1929 for measuring muscle fatigue. Hill's experiment tested frog muscles under 2 different sets of conditions. In the first set, the solution which was around the muscle prevented the diffusion of lactic acid, while in the other one, the solution allowed diffusion of lactic acid. Hill showed that when lactic acid could diffuse away from the muscle tissue, muscle performance improved significantly. It would work twice as long. Hill's conclusion went unchallenged for many decades. The explanation for the cause of muscle burn has changed over time as new evidence has accumulated. Does this represent a failure of the scientific method? No. The changing view of lactic acid's role in muscle fatigue illustrates an important point about the process of science. Scientific belief is dynamic and subject to constant adjustment as new evidence is uncovered. Organisms like yeast can survive with or without oxygen, and fermentation can produce different types of waste products which could vary from species to species. It could be ethanol, lactic acid, or other compounds. Oxygen is very important and it has an evolutionary connection. Both aerobic and anaerobic respiration start with glycolysis. Therefore, glycolysis is the universal energy harvesting process of life. Long time ago, billions of years ago, before oxygen was significantly present in the Earth's atmosphere, ancient prokaryotes used glycolysis exclusively to make ATP and generated ATP exclusively from glycolysis. This is the timeline of oxygen and life on Earth. The fact that glycolysis occurs in almost all living organisms is indicative of the fact that it evolved very early in ancestral life forms. Also, the location of where glycolysis occurs, it does not require mitochondria, it does not require any membrane-bound organelles. It just simply occurs in the cytoplasm. So this suggests that when prokaryotic cells were in existence, that is when it started.